All right. I think you can hear me without echoes. Hopefully you guys are doing okay. If you haven't been on Pronto, just to update you, the meiosis and the two reproductive lobsters are having a little bit of an issue. So they're skipping questions for, I don't know, whatever reason. So if it skips questions on you, don't worry too much, just do it one time. Okay, I'll go in and adjust next week once everybody's done with it. Okay. So any questions on lobster at all? Are you guys okay with that? Yeah. All right. Any questions at all about anything from last week? Are you guys okay with that? All right, hopefully you're okay. If not, then make sure that you guys post it on Pronto and then we can go ahead and help you, okay? Remember that you have your exam this week, right? You have exam, oh, you need to, there we go. You have a lecture exam, right? So make sure that you study blood, heart, respiratory. Okay. So steady, and you should do okay. All right. What do you guys have next week? What's the big thing that you have next week?
The lab exam.
Yeah, you have lab exam number 2.
Are we getting another study guide for that?
It's been up in the resources, so you can go there. But yeah, it'll also be posted in the week 15 module, but you can go to resources and you'll see a tab that's just for study guides. Okay, next week. So make sure that you study that study guide, right? And that you do the practice, right? I hope you saw the pattern last time. If you do the practice, then you're pretty set and pretty ready for the actual practicum, okay? So when you take the practice, should you use your notes or no notes? What should you do when you take the practice?
No notes.
No notes, yeah. Do it with nothing, because what you want to know is what areas you're struggling with, and then go study that, right? Spend your time wisely. So study effectively. And part of that means that you need to go and do practice problems so you can see where you're weak and then go study that. Okay, cool. It's going to be similar. So all multiple choice. And then you'll get your grade at the end of that. Okay. All right. Last announcement. I'm going to go ahead and skip for lecture. I'm going to end up skipping reproduction. Okay, we do it in lab already, so it's a lot of repetition between the lecture and the lab. So you already do it in lab, but we're not going to do it in lecture. Okay, so make sure that when you do the lab, you know it well. Okay, what I am going to do though is because the assignments are already up, I'm going to go ahead and do the, let me see, you guys are my 214. So you guys have a weekly homework assignment. So your reproductive homework, I'm going to make it as extra credit. Okay. So that should help boost your grade a little bit. Okay. So if you do the homework, it's extra credit. You don't have to do it. So it'll be optional. But it'll be extra credit if you do it. Okay. So I'll look and I'll see how I can do that, and I'll change the direction so you see how much it's worth once I figure it out. Okay. So that's a good option. Part of the reason I'm giving you this extra credit is because I had so many students that came in and asked for help this week, and they were working really, really hard. So they're really impressive. They've been growing in their exams. So I was really impressed that they continued, because man, this week 14, It's rough, right? We got like a few more weeks, so we're all in it, and everyone's working really hard. So that was great. And I also like that some of the students came in as pairs, so they're working together. They have a study buddy, they have their community. Okay. Oh, that's a good question. Will reproductive be on the exam? No. So I'm going to take it out of that last exam, and it'll all be on. I think you guys have renal system, right? So no reproduction on exam. Okay, but it will be in your lab exam. So either way, you still need to study it. Reproduction is a lot of, like, it's mostly female reproduction that you're learning, not so much male. For males, it's really just making a sperm and then maturing sperm, right? And then ejaculation and erection. Those are the three things. For assigned female reproduction, you're looking at menstrual cycle. That's a big one. Menstrual cycle. You're looking at ovulation. You're looking at what happens when your hormones change, right? So menopause. What was the other one? Labor and delivery, breastfeeding, right? There's a lot more when you add in a baby, right? Or trying to ovulate. So it's a lot of hormones on that, okay? So you'll see that in lab. I think you end up figuring out what happens when someone's trying to have a baby. So is the baby okay, right, genetically, or do they have an extra or missing chromosome? Those kinds of things, okay? All right, so just keep that in mind, right? You have your reproductive homework. It's going to be extra credit. I'm going to remove it from your exam. So you'll just have the renal chapter, but the renal chapter is hefty, right? You notice that we have 2 days for it because the renal chapter brings in almost everything that you've learned all semester, and it integrates a lot of the different systems into it, right? So you'll see respiration coming back. You'll see acid-base regulation coming back, buffers coming back, pressure coming back, osmolarity coming back, tonicity coming back, chemistry, it's all coming back. Okay, so all of that is going to come back. So when you're looking at renal, you really want to go back to your notes and review, right? Review those notes that you created, those short pages that just jog your memory so that you have a better experience learning physio for renal, okay? The first part here is mostly on GFR, okay? So it's about pressures and how we regulate blood pressure and how we filter, okay? The second part is really about fluid and electrolyte balance. So what's happening at the molecular level in the cell so that your pee changes different colors, different osmolarities, right? You balance your blood. So that's what we're going to be looking at. Your lab is really helpful. It's super helpful for the fluid and electrolyte balance. Okay, so all of those feedback mechanisms that you see in the lab, you're going to need to know in the lecture. And there's a lot of them. So just go and make sure that you really study that well this time around so that when you get to the lecture final, it's not that hard. Okay. All right, any questions? Let's see. I'm not sure what your question is, Abby. So if you can retype it a little differently, that would help. Oh, Maritza is asking if the extra credit homework is going to be multiple attempts. It'll be one attempt, but it's still open note, right? So just make sure that one attempt is good. Vanessa is asking, will the error be fixed on the lobster? Yes, so we talked about that earlier, and it's also on Pronto. So we'll see if they can fix it, but I really doubt it because it just seems like the responses I'm getting seems like I just have to go in and edit every single one. So that's going to be a hot minute before I actually go and edit all of those. Let's see. Next week, we'll just meet for lecture. No assignment, just the lab exam number 2. Yeah, so next week for lab, you're just taking the exam. That's it. And then the week after that, you're going to go ahead and do the last lab, and that's it. Half the class has a holiday and the other half has a lab next week on week 16. Okay. What else do we have? Yeah, do not retake the lobster. Nope, don't do that. Just take it once for this week. Okay, because it's having issues. It's not your fault, but we got to fix it. Okay. All right. Any other questions? Are you guys good? We're almost done. I feel like we're almost there. All right. So this is your chapter learning goals for renal. Okay. So notice that the first half is really about GFR, about pressures and blood pressure. The second half is really going to be about tailoring your urine. Okay. So feedback loops and mechanisms, excuse me, that help you change the chemistry of what the urine looks like when it comes out of you. Okay. And why that's important for you to maintain homeostasis. So a lot of the feedback mechanisms that we've seen, they're all going to come back. Okay. So let's start off with some easy stuff, right? Let's go over just anatomy again, right? I like to start off with anatomy so that you're not trying to remember all that anatomy as we go through, okay? So what are the kidneys used for? They help you regulate water, right? They help you regulate salt. So those are big ones. And really what they're helping you regulate is what's in blood, okay? So a lot of times students have the misconception that what you're regulating is urine, and urine is coming out of you, right? We don't care what's in urine. I mean, we care what's in urine because it affects what's in your blood. So we're really regulating our blood by using the renal system. Okay. Other things that it helps us regulate is things like our metabolic waste, toxins, and drugs. Right? That's why if they think that you're doing drugs, they're going to ask you for a urine test because that's where we're going to get rid of it. It also helps you with acid-base regulation, and that's a big one. Okay. So acid-base regulation is really important. Remember that your blood only likes to be between 7.35 and 7.45. So anywhere outside of that, you're going to kick in your renal system and your respiratory. So that's all going to come back. Okay. The last part here that sometimes people forget is that our kidneys make hormone. One of the things that it makes is erythropoietin, EPO. And that helps you with your red blood cell count, right? If you have a low red blood cell count, EPO can help increase it. Another one that we're going to see soon is renin. Okay, and renin will help you with blood pressure. Okay, so we'll see that in a few different feedback mechanisms. Last but not least, it helps you activate vitamin D. So if your kidneys don't work, you're going to have trouble getting calcium into your blood, right? And then you'll have bone issues, etc. So keep that in mind. All right, let's look at just the basic anatomy. Here on the right, you have a picture. You have normally 2 kidneys. Some people are born with 1. Your kidneys are connected to ureters. Ureters then take that fluid, they store it in the bladder. And when your bladder has stretched enough, then it triggers you to go to the bathroom, okay? So I want you to spend a few minutes and I want you to label the kidney and the nephron, okay? So I'm gonna give you 2 minutes to do this. And I want you to label what you remember, okay? Label the gross anatomy, which is on the left side, and then label the microanatomy and the nephron, and just label the parts that you know, okay? For us, because we're in physio, this by far is the most important part of the kidney for us, is the nephron itself, because that's how we're tailoring what's staying in blood and what's being removed from blood. Okay, so go ahead and take 2 minutes. I'm going to put my timer on and hopefully that helps. Okay. If you have questions, go ahead and unmute or type it. All right. So hopefully that was not too bad for you, right? And it's something that just comes right back. So I'll point to stuff and you tell me what you think it is. Okay. So what is this region called? So if you know, type it up. Good job. So this is the medulla, okay? So the main thing that you want to remember when you see the medulla is that it's salty, salty. Okay. So remember that the medulla is where you have the nephron going in, right? So it's where it dips down. And this salty, salty property of the medulla is really, really important because when we think about the osmolarity of our blood, right? We know that normally it's at 300 and the medulla is actually a lot more than 300. That is so important. You better know that, like forever, right? So the medulla is more than 300 milliosmolarity, and we'll see the gradient for that, okay? Good job. Yes, it'll go down up to 1200. So that's important, okay? So we'll see that gradient. The next part here is the top part. So this, what is that called? Good job. That's your cortex. And the thing that you want to remember about the cortex is that it's isoosmotic. So it's 300 milliosmolarity. Okay, you just have to know this. So these two numbers you have to just know. Okay, so forever know the cortex is 300, the medulla is salty, salty. Last but not least, you have, and I'm going to put here isoosmotic. Okay. Last but not least, you have right here a structure. Okay, when we look at this structure, we see that it has little holes in it. So what's the name of that structure with the little holes in it? So it's before the calyx. It's what separates the pyramid from the calyx. So this is your renal papilla. Okay, so when you look at the nephron, like what you see in the middle, right? You would see the renal papilla there. So this hole is the same as one of these dots. So you have a bunch of them. Okay. So the reason that's important is because everything that's above that hole is called filtrate. Okay, the fluid that's in the nephron is called filtrate. Anything past that hole is now urine. Okay, so the papilla is your boundary between filtrate and urine. The difference between those two terms is that filtrate, you can still change it and tailor it. Okay, we can change it and tailor it throughout the nephron. Once it leaves the nephron and it's in the calyx, we can't tailor it anymore. It is what it is, it's fixed. So urine is not tailored. Filtrate can still be tailored, okay? All right, next part here. What's the name of this tube? That one's pretty easy. What's the name of this tube here? So it's after the calyx. So this tube right here. Ureter, very good. Okay, what kind of muscle do you have around the ureter that helps you move fluid? What kind of muscle? Smooth muscle, right? That's why you don't have to think, right? When you pee, you don't have to think about it. You have smooth muscle around that will cause peristalsis to occur when you need to move fluid through, okay? So you still have smooth muscle that can move stuff through that ureter, okay? Okay, most of the time, urine will move in this direction, okay? Cool. So that's the goal. It's moving stuff from one to the other. All right, so let's look at the nephron. Let's look at the places that are important in the nephron. So here, from what is the name of this tube here? What is the name of this tube? The first tube that is squiggly. Let's see if you guys can see me. Good job. It's the proximal convoluted tubule. So this first part that has the first part, right, of the nephron, it's the PCT, proximal convoluted tubule. Okay. Do you remember what kind of tissue you find in that tube? Is it simple squamous or simple cuboidal? Oh, I'm pointing at the wrong one. Thank you. Oops. You guys remember if it's, good job, it's simple cuboidal. So the key here is anytime you see cuboidal, it's for absorption and secretion. Okay, so we're going to be absorbing and secreting a lot there. What about the second tube at the very end that's also squiggly? What is that called? Distal convoluted tubule, DCT. And that one also has simple cuboidal, so a lot of absorption and secretion happens there, okay? What about that tube that goes all the way down and comes back up? What is this called? The nephron loop or the loop of Henle? Okay, both are correct. Remember that you have two parts. You have the descending limb and then the ascending limb. Okay, so I'm just going to put loop. You have descending and ascending. So something to remember is that descending is going to go ahead and reabsorb water, and ascending is going to reabsorb salt. Descending reabsorbs water, ascending reabsorbs salts. Okay, so that's going to be important later on when we see this again. Okay. We'll talk about what allows it to move through as we go through GFR. Okay. All right. So the next part here that I want to go over is this yellow part that's going around these blood vessels. So what's that yellow part that's going around these blood vessels? What's that called? The glomerulus is the blood vessels. So this red part, this red, that's glomerulus. But what is the yellow part around that red part? Oh, good job. Bowman's capsule or renal capsule. Very good. Okay, so Bowman's capsule or renal capsule, both are okay. Remember, that's part of the nephron. Glomerulus is part of the circulatory system. Okay, so when we're looking at the Bowman's capsule, it's going around. Remember that it has these special cells called podocytes that surround the blood vessels. Those are going to come back to haunt us. Okay, and that's where we first make our filtrate. All right, last piece here that we want to remember is this tube right here. So what's the name of this tube going down to the papilla? We'll talk about those three layers too. Good job. Collecting duct. Awesome. Good. All right, so that's just the basic anatomy that we want to remember. Remember that a lot of this tissue is cuboidal, so you're absorbing and secreting throughout. Sometimes you'll have simple squamous, and that's for diffusion and osmosis. Okay. All right. So the next part we have here is about the nephron and the glomerulus, right? Remember, that's your blood vessels. So when you think about glomerulus, these are capillaries. And these capillaries are specific for filtration. So it's really, really important that we remember that they are fenestrated. Okay. So they're holy. They have little holes in them. The other thing that we want to remember is that blood will come in through the afferent arteriole, and then blood will come out through the efferent arteriole. So blood comes in, we have our glomerulus, blood comes out. So afferent arteriole and efferent arteriole, okay? The blood vessels inside the nephron, this is our glomerulus, okay? So blood in, blood out. The other thing that we want to remember is that the efferent arteriole is going to turn around and it's going to hug the nephron, okay? So we're going to see this efferent arteriole come back and it's going to hug the nephron, okay? And that's important because our blood is then going to be next to the nephron. So it's going to be next to the filtrate. And when you look at your book, some of the things that you notice is that they'll have arrows of things coming out of the collecting duct or the PCT or the DCT or the loop of Henle. And it seems like it goes into nothingness, right? You're like, oh, it's coming out of the nephron, but it has to go somewhere. And the place that it goes into is the blood vessels, right, that are hugging the nephron. So when you see an arrow coming out of the nephron, that means that it's going into the interstitial fluid and then it's going into the blood, okay? Things don't just kind of disappear. They always go somewhere. The assumption is that you know that it's being reabsorbed and being put back into the blood, okay? When the efferent arteriole turns around and hugs the nephron, depending on what it's hugging, it's going to be termed something different. So you have peritubular capillaries and vasa recta. Peritubular means around the tubes, right? The PCT and DCT. If it's the vasa recta, then it's around the loop of Henle. Cool, not bad. So make sure you just kind of review the anatomy. This part of the anatomy is going to become very important. So you need to recall this, okay? So this is the juxtaglomerular complex, okay? Or the juxtaglomerular, sometimes we call it apparatus. Okay, so when you're looking at the JGC or JGA, depending on what book you have, we're looking at three main parts. So the first part is called the macula densa. The second part is the granular cells, and the third part is extraglomerular mesangial cells. Okay, so we're going to break it down over on the picture that's on the right. So we're looking at this part here. Okay, you notice that we have some different parts of the nephron that are associated with the juxtaglomerular apparatus, right? So we have the Bowman's capsule over here. We have the glomerulus, which is our blood vessels inside the Bowman's capsule. And then we have the tube coming in and the tube coming out, right? The blood vessel coming in and the blood vessel coming out. So we have our afferent arteriole and our efferent arteriole. Right next to our afferent and our efferent arteriole, we're going to see another tube. Okay, so this is usually your ascending limb of the nephron, okay? So when you look at it, the key here is that the filtrate is passing by this structure, okay? So that's your main goal. The filtrate passes by this structure. And you have some specialized cells that connect these three structures here. Okay, let me pick a different color. So this right here is the most important part. So the connection between the filtrate, the blood coming in and the blood coming out, that's all important. So when we look at the tube that contains our filtrate, we have something called the macula densa. Okay, so that's these cells right here. They're right next to the filtrate. And so they're monitoring how much salt is in that filtrate, okay? The other thing that you're gonna notice is that we have cells right next to it. So we have our granular cells, okay? That's these purple cells here, okay? And granular cells are specialized smooth muscle cells, okay? So since they're smooth muscle, they're able to contract and relax, okay? And that's important. The other thing that's important to remember about smooth muscle is that they can have gap junctions in between them, right? So here, these smooth muscle cells have gap junctions next to them that can help them communicate with other smooth muscle cells that are next to them. So macula densa cells talk to granular cells, and they're able to contract and relax those blood vessels, okay? What else do we have?
I have a question. Can I ask a question?
Yeah.
You're saying that the macula densa detects the NaCl, so, and if it'll determine whether or not to open or close, or, you know, constrict or dilate the afferent. That's what they're doing. That's what they're there for. But they're doing that just by being next to them.
Yeah, so at some point today, we'll talk about autoregulation, and this will come back. We'll go through step by step what it does. Cool?
Okay, thank you.
Awesome. Yeah, cool. All right, some other things that are in here are the extraglomerular mesangial cells, right? So those are also smooth muscle cells that are connecting all the different cells. So we're connecting the smooth muscle cells around the blood vessels with the mesangial cells. And it's important to know that they're also smooth muscles, so they connect, right, with gap junctions. So those are the main parts, okay? The two parts that you want to remember the most are macula densa and granular cells, but all of this will come back, okay? All right, so now that we reviewed our anatomy, we can go ahead and move on to physiology, okay? The first thing that we want to figure out when it comes to the renal system is how we filter our blood, okay? That's our main job. How do we filter our blood, and how do we change that throughout the day when we eat something or we drink something, or how do other people regulate it when their physiology is not working normally, okay? So we're gonna go ahead and look at the first part. So the first thing is that we're always filtering, right? So we're always making urine. The place that we're gonna filter is gonna be in the Bowman's capsule. So our Bowman's capsule and our glomerulus are gonna be very, very important. This is where we first filter our blood, okay? So remember that the glomerulus has fenestrated capillaries. So we're gonna add blood into those capillaries. You have a high pressure in the blood, and that's gonna move it out into the Bowman's capsule, okay? We're always moving from high to low pressure. What you're gonna notice is that 100% comes in. And when we look at what comes out, right, it's 80%. So 80% is coming into that efferent arteriole. 100% comes in, 80% comes out. That means that that 20% difference is becoming filtrate, okay? And that's what we notice here. So 20% of our plasma goes into the Bowman's capsule, and when it goes into the Bowman's capsule, it becomes filtrate. But 20% is a lot, right? Imagine that throughout the day, you're peeing out 20% of your blood volume. You would end up in the hospital, right? So this is not efficient. What we need to do is we need to concentrate the things that we want to get rid of, and then bring back the things that we need. So if you notice in the very beginning in the proximal convoluted tubule, you're going to reabsorb about 19% of that filtrate, right? So you have 20% coming in, 19% of it's going to get put back in your body. So a little less than 1% is what you're going to lose. Okay. So you bring back 99% of that water, that salt, that sugar, those proteins, bring it all back. And the little bit that you need to get rid of will continue through the loop of Henle, the distal convoluted tubule, and the collecting duct. Okay, cool. All right, so make sure you remember that you're not peeing out 20%, but 20% comes out as filtrate initially. All right, we're gonna look at 3 processes. We're gonna look at filtration, that's where we start. Next time we come in, we're really gonna focus on absorption and secretion. So how do we get different items back into our blood, and how do we secrete them out of our blood, okay, throughout the different parts of the nephron. So let's look at what happens at the glomerulus, right? We know that we're getting 100% in, 20% comes out, and 80% goes into the efferent arteriole. So what actually filters our blood? So the first part that we need to remember is that you have different layers around that glomerulus, and those layers are really important for filtering. So we need to remember that we have fenestrated endothelium, right? That's your squamous cells that have little holes. That's the first part that filters your blood. The second part is your basement membrane. That's what's holding your epithelial tissue together. And that has proteins in them like collagen, right? So that creates a small microscopic net that helps you keep things in your blood, and other things get to be made into filtrate. Okay, so if you think about blood vessels, or sorry, blood cells, they're not going to be able to go through that fenestrated capillary. And if they do, you have a second net that prevents them from going through. If you think about proteins, right? Proteins will definitely be able to go through that fenestration, that pore, but they're going to get to that basement membrane, that microscopic net, and they won't be able to go through. Okay, so small proteins, yes, big proteins, no. And that's important because proteins in your blood maintain your osmolarity. And your osmolarity is everything. If you have a change in your osmolarity, you're going to suck water back into the blood, or you're going to push water out into the interstitial fluid, and that affects you. Okay. The third thing that you have, the third barrier, is the foot processes of podocytes. So remember that podocytes look kind of like little octopuses, right? And the way that the feet interact is by intercalating. They're doing this, okay? So when you have that, things can't go through to the other side. This is why if you see blood in your urine, that's a problem, because you have 3 layers of protection, right? All your red blood cells should stay inside your blood. They shouldn't come out to the other side. If they're coming out to the other side, that means you have enough pressure pushing these cells through the fenestrated epithelial tissue, through the basement membrane, and through the podocytes. Okay, you can imagine the kind of damage you have at the nephron in that microscopic level, and it takes a while to repair, if at all. Okay, right, there's a lot of no bueno, it's a lot. Okay, so make sure that you review again your anatomy, right? This shows you the different layers. So you have your footed processes of the podocyte. You're going to have your basement membrane in the blue, and you have your epithelial tissue in the red. Okay, the little holes here are your fenestrations. Okay, and things will move from high to low pressure, right? We have high pressure, low pressure, and things will move in this direction. Okay, so just remember, anything that's large, like red blood cells or large proteins, they should stay in blood. Small things like sugar, salts, like ions, water, they can go ahead and cross to the other side. Okay, not bad. Yeah, Brenda, sorry about the glitching. I'm getting on my end little signs that my Wi-Fi is not doing great. So sorry. Hopefully the recording's okay. But but I'm gonna try to write as much as I can, okay? All right, any questions about glomerular filtration and these 3 layers, or are you guys okay? Cool. So keep in mind that when we're talking about pressure, we're talking about hydrostatic pressure, okay? Okay, so that's what's helping us push fluid from one side to the other. There are other pressures that we're going to look at that are also involved here. Okay, so it's not just the amount of fluid that is in the glomerulus that moves it through, but that's the main force. Okay. All right. So when you're looking at glomerular filtration, okay, let me see. We're going to be talking about something called the GFR. Glomerular filtration, oops, rate. Okay. So GFR. Okay. And the GFR tells you the volume of urine, or sorry, of filtrate per minute. Okay. So it tells you a rate. How quickly are you making this? Okay. And one of the main things that affects it is this right here. It's called the net filtration pressure, okay, NFP. And so one thing that we sometimes make a mistake on is that we think that the net filtration pressure is the same as the GFR. This is not the same as the GFR, but they are correlated. Okay. So one pattern that you're going to see here is that if the net filtration pressure goes up, GFR goes up. If net filtration pressure goes down, GFR goes down. If there's no change, then there's no change. Okay. So you're going to see that they're directly correlated. Okay. So if one goes up, the other one goes up. If one goes down, the other one goes down. Okay. And this shows you all the different pressures that affect how you make filtrate. Okay, so when you're looking here, right, we want to look at all the different pressures. So we have HPGC, OPGC, and HPCS. Okay, so we're going to go through each one and we're going to talk about how they relate to your net filtration pressure and how that affects your GFR. Okay, so make sure, please make sure to study the definitions of these 3. Study them. Okay, so you have HPGC. This is hydrostatic pressure in the glomerular capillaries. So we're looking at how much blood, right, is coming into the glomerulus. Remember, when you think about hydrostatic pressure, think about a container, like a cup. The more fluid you have in that cup, the more pressure there is. The less fluid you have in the cup, the less pressure there is. So hydrostatic pressure in the glomerular capillaries is affected by the amount of blood that you have in the glomerulus. Okay, so this one is the main pressure that's pushing fluid from the blood through the filters and into the Bowman's capsule. Okay, so that's what we see here. So we see blood being moved into the filtrate. Okay, so plasma being pushed into the Bowman's capsule and it becomes filtrate. Okay, this is the main driving force. So if you notice, this one is 55 millimeters of mercury. Everything else is 30 or 15, right? So this is the main force pushing fluid from the blood to the filtrate. Okay, so it's about 55 millimeters of mercury. Think about your blood, right? What do you think would happen to this hydrostatic pressure if you doubled your blood volume? What would happen to the hydrostatic pressure in the glomerular capillaries? If you doubled your blood volume, what do you think would happen? Would it go up or would it go down? Good job. It would go up, right? You have more blood in your system, more blood's going to get into the glomerular, into the glomerulus. Okay. This is a good example of someone who's pregnant. If someone is pregnant, you double your blood volume, right? So pregnant people are constantly filtering their blood more than usual. So it's not just, oh, the fetus is pushing on the bladder. That's part of it. The other part is you have double the amount of blood that you normally do. So you have more pressure pushing fluid into your Bowman's capsule. Okay. So keep that in mind when you see a pregnant person. You're like, oh, I know what's happening in there, right? And why you have to pee all the time, or part of the reason. Another thing that can happen when your blood volume goes up is that blood pressure does what? What happens to blood pressure? Does it go up or does it go down when blood volume goes up? It also goes up. They're correlated, right? If blood volume goes up, blood pressure goes up. So your blood pressure really affects the hydrostatic pressure in the glomerulus. If your blood pressure goes up, the hydrostatic pressure of the glomerulus also goes up because that's what's driving the fluid to go in here. Okay, so your blood pressure is really important. People that have a high blood pressure tend to do what all the time? What are they constantly doing? They're going to the bathroom, right? They're peeing all the time because their hydrostatic pressure of the glomerulus is higher than normal. So they're constantly making filtrate. Okay, so keep that in mind. So as you learn, pathology, keep all of that in mind. It comes back. Okay. All right, so let's look at the next pressure. So you know how much pressure is pushing fluid out. There's another pressure called the hydrostatic pressure in the capsular space. Sometimes it's called the hydrostatic pressure of the Bowman's capsule or the renal capsule, okay? But just remember that it's this fluid here, okay? That's what they're talking about. And remember, the Bowman's capsule is also a container. So the more fluid you have in that container, the more pressure you have. The less fluid in the container, the less pressure you have, okay? So when you're looking at that pressure, this is pressure that's pushing fluid from the Bowman's capsule back into the blood. And usually you only have a little bit of fluid there, so the pressure is small. It's, in this case, 15, right? So you see 15. So you have 15 pushing out, 55 pushing out, you have 15 pushing in, okay? The next one here is called the colloid osmotic pressure. And that's another one that I'm putting hearts next to. That one's really, really important. Remember that colloids are floating in a solution, okay? So think about the things that float in your blood. One of the main things that floats in your blood is protein, okay? And one of the main proteins is called albumin, okay? And albumin is really important to monitor because it's the one that really affects your colloid osmotic pressure. And the more stuff you have, the more albumin you have, the more it will suck, right? So you want to remember solutes suck, and they suck water. So when you're looking at your blood, right? I'm going to put albumin here in little dots, right? You have all these little dots inside the blood. And those little dots, those little albumins are solute. The more solute I have there, the more I pull fluid back into the blood. Okay, so imagine that someone has a lot of albumin in their blood. Are they going to pee a lot or a little compared to a normal person? If someone has a lot of albumin in their blood, will they pee a lot or a little compared to a normal person? Good job. They're going to pee very little, right? Because a normal person is pulling a little bit of blood or a little bit of solution back into their blood. But someone with a lot of albumin, a whole lot, they're going to pull back a lot more solution into their blood compared to normal. So it's going to come back to blood. It's not going to become urine. Okay. So it's important to know what someone's albumin concentration is, because that tells you how they're regulating their blood volume. Okay. What would happen if someone has very little albumin? What if they have very little albumin? How much filtrate will they make? Will they make more filtrate or less filtrate than normal? A lot more. Good job. So instead of pulling fluid back into their blood, they're letting it go. Okay. Remember, solutes do what? They suck. So if you have a little bit of solute, you have a little bit of sucking. If you have a lot of solute, you have a lot of sucking. Okay. And that affects how much urine you make at the end of the day. Okay. All right. So you have all of your different, let me put this up here. All of your different pressures. Okay. Oops. Solutes. Suck water. Okay. So let's blow up this equation down here. So you have net filtration pressure equals the hydrostatic pressure of the glomerulus, right? Minus the hydrostatic pressure of the capsule, right? So how much fluid there is in the Bowman's capsule, plus the osmotic pressure in the glomerulus, okay? So when you look at this, it's just basically saying pressure pushing out and this is the pressure of sucking. Okay, so sucking fluid back into the blood. Okay, you have pressure pushing out of the blood and then pressure of sucking it back into the blood. Okay, so at the end of the day, we want to know how much total is going to be pushed into the filtrate. Okay, so we're going to have 55 minus 15 plus 30, and that gives you 10 for your net filtration pressure. Okay, so because it's a positive number, if it's positive, then you are making filtrate. Okay, the higher the number, the faster you're going to make filtrate. The lower the number, the slower you're making filtrate. Okay, does that make sense? And then remember the relationship. Net filtration pressure is related to GFR. If NFP goes up, right, there's more pressure to make filtrate. That means that the rate that you're making urine goes up. Okay. Any questions about net filtration pressure or GFR?
I have one.
Yeah.
I don't think I caught on to the part where it says hydrostatic pressure in the capsule, which is filtrate pressure of 15 mmHg. That's the one going back
That's this
in,
one.
but that's like blood, right? That's like just blood coming back in to the capsule.
That's fluid going back into the blood.
Fluid going back into the blood, like
yeah, the same with osmotic pressure, right? Because remember, you have those three barriers, right? You have your fenestrated capillaries, the basement membrane, and the podocytes. So those three should keep red blood cells and big proteins in blood. And what can move through is things like ions, glucose, and water. Okay. So when you see these arrows moving back and forth, that should only be ions, sugars, and water.
Okay, so the bottom one, the HPCS, is like fluids, sugars, and water going back into the.
All three arrows, all three of them that you see there, they represent sugars, ions, and water. Okay.
Okay.
And so what you're looking at is pressure. So are you pulling fluid back into the blood, or are you pushing fluid out of the blood? And we have one that's pushing out and two that's pushing back in. Okay.
All right. Thank you.
Not bad? Yeah. All right, so let's take a look at how you regulate this, right? So we had a few examples just by thinking about it, okay? So we looked at like something like albumin and how albumin affects it. Think about other things like sugar, right? Who would have a high amount of sugar in their blood? So we looked at albumin, but what about sugar? Who would have a high amount of sugar in their blood? Diabetic
One who's
patients,
diabetic.
right? So when you look at someone that's diabetic, right, you're thinking, oh, they're constantly peeing, right? But one of the things that we're going to see, right, with sugar is that sugar is not large. Sugar is small, right? So when you think about sugar, sugar is able to go to the other side. So when you have sugar in your blood, you're going to have a lot more sugar in your filtrate. Okay. And that's going to affect how everything works down the line. Your proximal convoluted tubule, your loop of Henle, your distal convoluted tubule, and your collecting duct. Your albumin doesn't cross. So it's not affecting your filtrate, but it is going to affect how your blood that's coming around, right, through the peritubular capillaries and the vasa recta, it'll affect how they work. Okay. So as you go through, we're going to come back to this picture mentally, right? So just keep it kind of handy so that you can go back to it. Okay. All right. So let's look at GFR and how the GFR is working. Okay. All right. So this is your glomerular filtration rate. This is the volume of filtrate per minute by both of your kidneys, okay? Remember that GFR and net filtration rate are related directly. So if one goes up, the other one goes up, okay? We have different things that affect it. So number one, right, is surface area. Okay, another thing that can affect it is the barriers, okay, and the permeability of those barriers. So imagine that you have scar tissue in your kidneys, okay? It's going to be harder to filtrate because you have all these extra collagen fibers with this barrier, right? So permeability is going to be harder. If you injure your blood vessels, your glomerulus, then when they heal, you might have your fenestration be larger or smaller than normal, okay? And that affects it too. So let's look at some things that can affect it, okay? So your goal here is to maintain glomerular filtration rate in the kidneys. You want to keep it consistent, okay? And we have something called auto-regulation to help us maintain our GFR. And that's the key thing that we want to know for our exam, okay? We want to know how we auto-regulate. The other thing that can affect our GFR is blood pressure. Okay, so remember that GFR is making filtrate, and filtrate is coming from plasma. So if we make a lot of filtrate, that means we are removing a lot of plasma. If we make a little bit of filtrate, we're removing a little bit of plasma. Okay, and that's important because if we are creating a lot of filtrate, our blood volume goes down and our blood pressure goes down. If we keep a lot of our fluid in our blood, then we make a little bit of filtrate, our blood volume stays high and our blood pressure goes up, okay? So think about someone who has high blood pressure. One of the things that we can give them are water pills. And that's important because if you give them water pills, they make more filtrate. That's the main job. Let's make them pee more. And if they pee more, we lower their blood volume, and that helps lower their blood pressure. Okay, so blood pressure and GFR are very related. If one goes up, the other one goes up. If one goes down, the other one goes down. Okay, so keep that relationship in your mind. Okay, so the goal of GFR and blood pressure, that relationship, is to maintain the blood pressure in your system. So one of the ways that we can maintain your blood pressure is by changing the GFR to either maintain fluid or remove fluid from your blood. Yes, so if you make a lot of filtrate, you tend to pee more, okay? And you'll see why I say tend, because remember that you still have all those other tubules that help you reabsorb more fluid, more salts, more sugars, more proteins, okay? So we'll see that as we go along. But usually if you make more filtrate, right? So if your NFP goes up, your GFR goes up. And if your GFR goes up, blood pressure will go down, okay? So let's write that down, right? So if you increase net filtration pressure, then you increase GFR. And if you increase GFR, you tend to lower blood volume. And that lowers blood pressure. Okay, and then vice versa. So if net filtration pressure goes down, GFR goes down, it increases the blood pressure or blood volume. And here, when I say increase, I mean maintains, right? Because the only way to increase your blood volume is by drinking water, okay? And this will help you increase your blood pressure, okay? All right, so we're going to be looking at autoregulation, and next time we come in, we'll look at extrinsic controls for blood pressure as well, okay? All right, so let's take a look at autoregulation. And let me see if my slides change. There you go, awesome. So remember, these are intrinsic controls, so this is all local, okay? So think about local. When you think about autoregulation, we're really going back to that juxtaglomerular apparatus. Okay, so we want to go back to this area that we see right here. Okay, so let's go through and figure out why you would want to do this. Okay, so remember that one of the main goals that we have is to maintain a constant GFR. And the reason that's important is because we want to maintain our blood pressure pretty constant. Okay, so MAP, mean arterial pressure, that's blood pressure. Okay, and we want to maintain it within our own normal range. Okay, autoregulation will help you with that so that you keep that GFR pretty normal. Okay, so you're always making the same amount of filtrate on a regular basis. Okay, too much, then your blood pressure would go down. Too little, then it would go up too much. So we're going to look at two mechanisms. We have myogenic, and remember, anytime you hear myo, that means muscle. And the second one is tubular glomerular feedback mechanism. So the tubes and the glomerulus are going to work together to create this mechanism. Okay, so the first one here is myogenic response. So this is your smooth muscle. Remember that smooth muscle will react whenever it's being stretched, right? A really easy example is when you swallow, right? When you swallow, that begins peristalsis. Here you have smooth muscle, right, around those blood vessels. So one of the things that can happen is that they can change to respond to a change in pressure. Okay, so if there's too much blood coming in, that could cause the capillaries to burst. Remember that capillaries are just one simple squamous layer, right? So too much pressure will cause them to burst, and then the nephron is not going to work the way it should. So one of the ways that you protect it is by constricting if there's too much pressure, okay? So your arterioles will auto-regulate to try to maintain the same net filtration pressure that will help you maintain your glomerular filtration rate. Okay. It also prevents injury. That's the main thing. You don't want to injure your glomerulus. Okay. Any questions on myogenic response? That's the first one. The other one, the more important one here, right, includes your juxtaglomerular apparatus. Okay. So this is local control, and we use paracrines. Remember that paracrines are chemicals that get released out and they talk to their neighbors, okay? So this is important because you have the juxtaglomerular cells that are all really next to each other, and they're easy, it's gonna be easy to communicate from one place to the other, okay? So when we look, we have two types of cells that are really, really important. The first one is macula densa, the second one is granular cells. So when we look at the macula densa, these are cells that are right next to the tubules, okay? And they're constantly monitoring the amount of salt in the filtrate, okay? The more salt you have, the more filtrate you tend to make. The less salt that's passing by, the less filtrate is passing by, okay? More salt, more filtrate, less salt, less filtrate. And the macula densa cells are just constantly monitoring how much salt is going through, okay? The granular cells have their own job. So granular cells, remember, they're specialized smooth muscle cells, and they're gonna be able to release out an enzyme called renin, okay? And renin is gonna be really important in regulating your blood pressure and your blood volume, okay? So keep this enzyme in mind. So let's go ahead and see how this works. Okay, so we're going to look over here at this mechanism. So the first thing that happens is you have an increase in your glomerular filtration rate. So this goes up. Okay. When you have an increased GFR, that means that you have more filtrate coming into the Bowman's capsule, more filtrate in the PCT, more filtrate in the loop of Henle, okay? And distal convoluted and collecting duct, okay? More everywhere. So what happens is that your tubules are gonna have more filtrate, and your macula densa is gonna sense more salt passing by. And that tells it, hey, we're making more filtrate than normal, okay? When that happens, they're gonna go ahead and release a chemical, right? And that chemical is gonna go and it'll talk to the neighboring cells, okay? If there are other neighboring macula densa cells, it'll tell them, hey, release the chemical too. They'll go and they'll talk to the cells next to them. Some of the cells that they talk to are gonna be arterial cells. So they're gonna talk to the smooth muscle cells around the arterial, the afferent arterial. Remember that blood is going to the glomerulus through that. So it's gonna tell it, hey, we're getting too much blood coming in. Can you go ahead and constrict? And those smooth muscle cells are gonna constrict, and it's gonna go ahead and constrict the arterial, okay? So resistance goes up, less blood is going into the glomerulus, and your hydrostatic pressure of the glomerulus is going to go down. So when that happens, you're going to have a decreased net filtration pressure, and that's going to cause a decrease in your GFR. Okay. Anybody lost on that? Cool. Awesome. So make sure you know those relationships, right? The more filtrate you make, the higher your GFR, the less filtrate you make, the lowered GFR, okay? So all of this is just to maintain that GFR consistent. So let's go ahead and take a look over here at autoregulation and how that works, okay? So when we look at autoregulation, it helps you maintain that GFR within a specific range, okay? So we have our GFR here on the y-axis, and we have our mean arterial blood pressure on the x-axis. And when we look at our mean arterial blood pressure, this is our normal, okay, between 80 and 180. So that's our normal systolic blood pressure. And you notice what happens for GFR when you're within that range, right? We're able to auto-regulate so that we are normal within those normal ranges. If our blood pressure gets too low, then our GFR starts decreasing. If our blood pressure gets too high, then our GFR starts to increase, okay? But our body likes to keep it pretty consistent all the time through autoregulation, okay? All right, any questions on this slide? Are you guys good? Good. So let's look at extrinsic controls. These are going to be hormones and also nervous system. Okay, so we're looking at hormones and nervous system. And we'll see these again. So this is just an intro. This is all going to come back. Okay, so part of your extrinsic control is to again try to regulate the GFR so it stays about the same. And that helps you maintain your blood pressure, okay? So the whole thing here, right, for both of them is maintaining your blood pressure, maintaining your blood pressure. You don't want it too high or too low, okay? And GFR helps you with that. We're able to affect lots of different things in our body to help us regulate that blood pressure and that GFR. So when we look at nervous system, we can think about things like our sympathetic nervous system, fight or flight, right? When that happens, then our blood pressure goes up. That's going to affect how our GFR is working, right? If we have parasympathetic, that's also going to affect it. If we have norepinephrine given to us as a drug, that's also going to affect it, right? So you have all these different things from the nervous system that can affect you on how you create your GFR, okay? So if we think about sympathetic nervous system, you tend to dilate your blood vessels. This also includes your renal. Your autoregulation at the GFR or at the glomerulus is gonna be the main thing that prevents those capillaries from bursting. Okay, so what we just talked about like a minute ago, that's what's gonna make sure that the pressure in the glomerulus is not too high to protect your nephron, okay? Other things that can happen is if you have low extracellular fluid, right? So imagine low blood pressure. That could also affect your GFR because now your pressure is low, your net filtration pressure is also going to be low, and that causes you to have less GFR, okay? The other thing that we're going to learn a little bit later, and I'm going to put a whole lot of stars or hearts next to it, is the renin-angiotensin mechanism, okay? So this is called the RAS system. Okay, so we're going to see this later, but this is a network of a lot of different hormones that work together to regulate your blood osmolarity, your blood pressure, your blood volume, okay? And all of these hormones get secreted into your blood, and they're going to cause different systems to change how they are to help you regulate that GFR, okay? So this one will all come back, I promise, okay?
I have a question.
Yeah.
The extrinsic controls, that's like, is it safe to say that these come into play like if someone isn't healthy, or is it happening like all the time, just not just when you're not healthy? Or is this just like a
That's
when you're healthy?
That's a good question. When you think about intrinsic, think about within the system, right? So local. When you see extrinsic, think about outside that system. So here we're not only looking at the kidney, right? We're looking at all these other systems in your body that work together to help your kidney do its job, right? So it's not just local, it's everything else outside of it. So nervous system, heart, lungs, hormones, right? They're all coming into play with intrinsic control. Does that make sense?
Yes. Okay.
So don't get too caught up with intrinsic and extrinsic. Instead, really think about how would sympathetic nervous system activation affect my GFR? how would the RAS system affect my GFR? Okay. And we'll see both of them again. This one will come back with baroreceptor. Oops. Reflex. Okay. So this sympathetic nervous system, it'll come back with baroreceptor reflex. So we'll see it again. Both of these we'll see over and over, especially not tomorrow, next week when I see you again. Okay. So this one's coming back.
Thank you so much.
Oh yeah. Everything that we say today, everything, it's all going to come back next time. So make sure that you really study, make sure you go and you review your respiratory, your cardiovascular, your hormones, like go review that, because it's all just going to come back. And if you review it, it doesn't feel as fast. But if you don't review it, then this is going to feel really fast. Okay. So real quick, just to go over what we kind of learned already, right? This chart is really nice because it shows you GFR regulation. It shows you what we learned already locally, and then what we're going to go into. Okay. So for example, over here, you see a feedback loop for blood pressure. Okay. This shows you what happens when you have low blood pressure. And you notice that there's 4 different colors here. On the left, you see what happens with myogenic mechanisms. This is auto-regulation, your smooth muscle in your afferent arteriole either opening up or closing up, okay? And this shows you how it helps you regulate your blood pressure. The other mechanism that you have is your juxtaglomerular apparatus. This is your tubular glomerular mechanism. This also will affect how you either open up or close up your afferent arteriole, right? And how much filtrate you're making. So you have these first 2 helping you regulate your blood pressure. The next part here, you see how the RAS system affects your blood pressure. Okay, you notice that we see a lot of systems. We see our endocrine system, our blood vessels, our kidney, right? This also is going to include your heart, right? But you see a lot of systems coming in for the RAS system that help you affect your GFR, that helps you affect your blood pressure. Last but not least, you see neural control. This is your sympathetic nervous system specifically, how you change that tone in your sympathetic nervous system that will affect how much your GFR is producing, and then that affects your blood pressure. Okay, so you can see how everything kind of comes into play. So for example, you see this arrow here. So what happens in one regulation affects the other, right? The same here. So what you're noticing as we go through this chapter is that you're putting the puzzle pieces together, right? It's not memorize one mechanism and that's it. You have to see how those mechanisms fit together, okay? All right. Let's see if this likes to. There we go. So let's try again. There we go. That's not it. Oh man. There we go. Cool. All right. And then I don't remember what your name was, but this is where you have the definitions between intrinsic and extrinsic. Okay. All right. Any questions about GFR, how important it is, NFP, how important that is, and the basics on how blood pressure, blood volume, NFP, and GFR, how their relationship is, or are you guys okay? Good. All right. So we went through just the Bowman's capsule. Okay. What we're going to go into next is what happens in the tubes. Okay. So remember that in the tubes, you have cuboidal epithelial tissue, and that's meant for absorption and secretion. Okay, so we're going to look at how it does that. Okay, so the main patterns of absorption, the main patterns of secretion. So we're going to start off with tubular reabsorption. Okay, so I want you to kind of think about that word reabsorption. Okay, so it means absorb again. Okay, we're absorbing these things again. The first time we absorbed it was in the digestive tract. Then we put it in our blood. Then we went ahead and put it into the filtrate, and we're like, oh, no, 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 we want to keep it. So let's absorb it again, put it back into the blood. And that's why we call it reabsorption. Okay, so it's the second time that we're putting it back into the blood. When we look at reabsorption, what we want to remember is that it's either going to be active or passive. Okay, active means we're going to use ATP to power the movement from one side to the other, or we can use secondary active transport to help us move it from one side to the other. The passive reabsorption is diffusion and osmosis, right? So again, remember, solutes suck. That's our main thing. Solutes suck. And things will move from high to low concentration. Okay, so keep those patterns in mind. So we're going to be looking at two main processes. The first one is called transcellular, and the second one is called paracellular. So trans means across. You're going through the cell from one membrane to the other membrane and coming out the other side. Paracellular means that you are going between cells. So this one is through cells, and this one is between cells. Okay, so think about water and ions. They can go through in between those cells. Okay. So let's look at these two routes, and then let's break it down in the figure that you see here on the right. Okay, so we have transcellular route and we have paracellular route. Okay, when we look at the figure on the right, I want you to think about the ions, the water, the material that's moving through these two routes. Okay, so the first one here, you have water and solutes. Okay, so let's look at water first. Can water diffuse through the cell, right? So through from one side to the other side. And the answer is true, right? Remember that water can passively move through membranes unless you're told otherwise. So water can go through one membrane and then through the other, as long as it's being sucked to that area. It can also go through paracellular route, so between the cells. It can go between, it's small enough, right? It can go through those tight junctions. The next thing that we need to see is something like a solute, right? So when you think about solutes, think about salts, small proteins, what else? Salts, proteins, some drugs, right? It just kind of depends on what we're looking at. Some fats. These different solutes, they can go through transcellular route, right? And things that are big or charged or that can't dissolve easily through the membrane, they can go through transcellular transport. So those items can go into a vesicle. That vesicle can be transported from one side to the other side, okay? If they like fats, right? So think about things like phospholipids, right? Then they can move from one membrane to the other membrane a little easier, right? So solubility matters. If they're soluble through the membrane, it's easy to move them through. If they're insoluble, then you can use vesicles, okay? Paracellular route, these have to like water. So they tend to be things like ions, sodium, chloride, calcium, and water. They can move through. They're small enough, they're charged, but they can definitely move through. If it's a sugar, it's going to have to go through transcellular transport because it's a big molecule compared to an ion, okay? So if we look here, transcellular route, right? They have to go through from one membrane to the other. And then when they get released, they get to go into the blood. Paracellular, think small, water, calcium, magnesium, potassium, sodium, okay? They can definitely move in between the cells, okay? They have to go through tight junctions, so very, very small. At the end of the day, everything ends up in the peritubular capillaries, right? So we're in the PCT and we move material back into the blood. This material is reabsorbed. Solutes or water, okay? So that's key. So 2 ways to transport depending on who you are. If you're large or if you're charged through the cell. If you're small, then you can go paracellular, okay? Lipids are nonpolar. Good. All right. So the other way that we're able to transport items is through secondary active transport, right? So when we think about secondary active transport, usually we have one protein that is making a gradient and another protein that is using the gradient, okay? Primary active transport is going to be the protein that is using ATP directly, okay? So here we have an example. Here we have our sodium potassium pump, right, making a gradient. So over here you have sodium being pushed out, potassium being pushed in, okay? So remember, you always have these everywhere and they're constantly making these gradients, right? You assume that you have these gradients no matter what. This gradient is going to be important because it's going to cause you to have a low amount of sodium inside and a high amount of sodium outside, okay? So that gradient is important. So we're gonna have a high amount of sodium on this side, a low amount inside that allows sodium to go from high to low. And that's important because a lot of different solutes can use that to be transported through the cell. So glucose is a really famous one. When we absorb salt, we reabsorb salt, we reabsorb glucose. Other things are amino acids and some vitamins, okay? So this is secondary active transport because sodium gradients are powering the movement of these solutes through the membrane, okay? Good, I think we're good with that one, all right? So keep this in mind. You have sodium, potassium pumps that power many of these secondary active transport proteins, okay? So things like glucose, amino acids, and vitamins. Okay. Other things that we can use to move items across from the filtrate back into the blood are passive tubular reabsorption. And in this case, we're really looking at water. These are your aquaporins. Okay, aquaporins, remember, these are these channels that you have everywhere. And that's why water is able to cross the membrane all the time. Okay, in your PCT, you have aquaporins, and we can actually change the amount of aquaporins that are in your nephron. So here we notice that we have our aquaporins, and then water is able to move from one side to the other side, going through those aquaporins. The more aquaporins you have, the faster it can move from one side to the other, okay? All right. The other thing that we have here is passive tubular reabsorption of solutes, okay? So some solutes are going to be able to cross the membrane passively. So things like lipid soluble substances, right? So if they're nonpolar, they can cross the membrane and it's just passive. It just crosses the membrane, goes to the other side, crosses that membrane, then goes into blood. Okay, that's also passive because you don't need to power the movement through the membrane. Okay. All right. All right. Any questions about the different kinds of transport? Or are you guys okay so far? All righty. So This one, make sure you know really well, okay? So we have seen this before. So we're seeing it again, but now we're seeing it in the renal system, okay? So this is the sodium glucose transporter, okay? Sodium glucose transporter, okay? SGLT. And this one requires you to have the sodium potassium pump active, okay? So when you look at the sodium potassium pump, you notice that it's on the basal side, okay? So on the basal side. And you're making your gradient, okay? So at the end of the day, when we look at this gradient, we're gonna have a low amount of sodium inside and a high amount of sodium outside. And that gradient is important because when we look at our filtrate, you have a high amount of sodium on the outside of the cell and a low amount inside. So it's going to move from high to low concentration. Okay. Glucose is going to be a co-transporter. So when you have glucose and sodium bound to the SGLT, it will cause it to move into the cell. And this is how we get our glucose and our sodium in. The sodium will get pumped out from the sodium potassium pump, and then the glucose can be moved through a glucose transporter on the basal side, and it'll move from a high glucose concentration to a low glucose concentration. Okay, so it'll move from high to low. Not bad. So the sodium potassium pump allows us to move glucose into the cell. That creates a high amount of glucose in the cell, and then the glucose transporter can move it from high to low out of the cell. Okay. Remember that once it's in the interstitial fluid, it's going to get reabsorbed into the blood. Okay. So it's going to go into the peritubular capillary and it's going to go back into the blood. Okay. So when you guys look at this, right, do you think that you can reach saturation? So can you reach saturation? Another name for this is transport maximum. And we saw these relationships earlier, right? Earlier in the semester. Good job, Karen. Yes, you can definitely reach saturation. You can reach transport maximum. You only have so many glucose, sodium glucose transporters in your proximal convoluted tubule. You only have so many. Once 100% of them are working, you reach saturation. You cannot reabsorb any faster than that. So if you add more glucose to the filtrate, you're not going to be able to pick it up again. So where do you think that sugar goes? If it's staying in the filtrate, where am I ultimately going to see it? Good job. Yep, you're going to see it in the urine. So how is your urine going to smell if you've reached saturation? What's it going to smell like? Sugary. Yeah, it smells like juicy fruit gum. That's like the best smell, or like a pear. Some people say it smells like a pear, but it's going to smell sweet, right? And usually if you notice that your urine is smelling sweet, then you should probably go to the doctor and see if maybe you've developed diabetes, right? So when you've reached saturation, you have so much sugar in your blood that you're going to have a lot more sugar in your filtrate, and your proximal convoluted tubule can only work so quickly, right? So if all of the SGLTs are working, and you still can't pick up all the glucose, then that probably means that your sugar is not regulated, okay? So people with diabetes tend to reach this saturation point. Okay, so think diabetes. Okay, or you have too much sugar. Okay, so you're going to find that they have that sugary urine, and then you can go, and if you regulate your sugar, then that'll regulate how much work your PCT is doing to bring back that glucose. When you look at urine should have no glucose in it. If anything, just trace amounts, okay? But you should have pretty much none because you're not reaching saturation. You should bring them all back. And glucose is something that is so special for you because we use that in our brain, right? Our heart likes to use glucose. So your brain is the main thing. You want the glucose because your brain likes glucose. It doesn't like other stuff. So you try to keep your glucose as much as you can. All right. So when we look at the data for someone that has reached transport maximum, this is what we're looking for, okay? So here on the right, let's look at the figure. You have your plasma glucose. So this is your blood. And then you have the glucose filtered load. Okay, so reabsorption or excretion, depending on which one we're looking at. So how much is filtered, the reabsorption of it, or how much we're removing out, okay? Because we have different lines, we're gonna have different y-axes, okay? Maybe I'll color code them with what's here. So let's look at the first one, okay? So we're gonna look at the blue line. So we're looking here, okay? I want you to notice what normal is. So normal is gonna be between 0 and about 300 plasma glucose. Okay, that's normal, between 0 and 300. And if you're between 0 and 300, then you're gonna notice that you're not gonna have a lot of glucose in your urine, right? You're gonna be having a little bit. So let's look at excretion. How much sugar is in my urine? Okay, this is what's in urine. So when we're between 0 and 300, right, you're at about 0, right? You start going up a little bit at around 200 and 300, right? You start to go up a little bit here, but you're still pretty low. Compare that to 500. You're going to have a lot more sugar in your urine. If you get to 700, even more, right? You're getting more sugar. So the more sweet it smells, the more sugar you have in it, okay? So let's look at the reabsorption, the blue, okay? So we have 0 and 300, okay? Between 0 and 300, you're reabsorbing most of it, right? So you're putting in sugar, but you're reabsorbing a lot of it, right? You're keeping up with it. That's why you're moving up. You're keeping up, you're keeping up, you're keeping up. And at around 300, you notice that you level out. When you level out, this is your maximum, okay? So once you reach this point where you're leveling out, you've reached your maximum, right? You cannot keep up anymore. You can add more sugar, but your PCT cannot keep up, okay? So that's why you notice that your excretion starts to go up. So when you reach transport maximum, you start to excrete more sugar in your urine because you just can't keep up. So you're gonna pee it out more, okay? Good question, Tatiana. She's asking, would someone with renal failure also have lots of sugar in their urine? So if someone has renal failure, they're struggling to make urine. So their nephrons are not working the way they should. So the urine that they do make, you have to kind of check to see if they're reabsorbing and secreting normally. So that's part of the process as you go through, and that's why you change your diet with different kinds of renal failure as you go through, okay? It'll look different depending on where they are and what they're eating and also all these other things, right? So remember that big feedback mechanism in the beginning with the GFR, and you saw how you had 4 different colors and a lot of things coming together. That's part of the reason nephrologists are highly regarded, right? Because you're putting in all of these systems together that will affect how your urine looks, that mirrors what's happening in your blood, and your blood goes everywhere. So it's affecting a lot of organs, your heart, your blood pressure, etc. Right? So it'll be different depending on a lot of different things. Okay, but we'll see renal failure a few times along the way as we do case studies. Okay. All right. So we have those two. Those are the main ones. And we talked about diabetes with this, right? All right. Cool. That's here. All right. So let's take a look at the next part here. So the next part here is reabsorption, not only in the PCT, but in other parts of the nephron. Okay, so we've looked at PCT along the way. Okay, one of the things you want to remember about PCT is that it's going to help you reabsorb a lot of that sugar. Okay, so remember that a lot. Okay, so PCT really helps you regulate that sugar. Okay, it reabsorbs it. Other things that you see here is amino acids, vitamins, and then some special ions. Look at those ions, right? They're the ones that we love. Sodium, chloride, potassium, right? It's those four that matter. So sodium, chloride, potassium, calcium, and then magnesium. But those ions are highly, highly regulated, and your PCT really helps you maintain them, right? They're hard to get, so you want to keep them. Water is another one. Remember that 20% of that plasma goes into the filtrate, and your PCT reabsorbs 19% of that fluid. That includes a whole lot of water. So your PCT is awesome, okay? Some lipids and then urea, okay? But make sure you know these highlighted ones for PCT, okay? The other ones that we're gonna look at are right below. So the next part that we're gonna talk about is the nephron loop or the loop of Henle, okay? Some key things that you wanna remember there is that it's all about water and solutes, okay? So we're gonna look at reabsorbing a lot more of that water. So that 1% that gets there, we're gonna absorb a lot of that water back because we don't wanna lose it, we don't wanna dehydrate. And the second thing that we reabsorb is ions. Okay, these ions in particular, so sodium, chloride, and potassium. Okay, and if you look at the loop, remember that it is in the medulla, right? And remember the medulla is salty, salty. So if we look at the medulla and you had to determine what salts are in the medulla, these three are the ones you're going to see the most: sodium, chloride, and potassium, because those are the ones that we push into the medulla to make it salty, and that helps us pick up the water, okay? After we go through the loop of Henle, we're gonna go ahead and look at the DCT, okay, the distal convoluted tubule. It's also gonna help us regulate and tailor our urine a little bit more, so we're really picking up more ions. And our favorite ones are coming back, right? Sodium, chloride, calcium. Last but not least is the workhorse of tailoring your urine, right? Extra special is your collecting duct. Okay, so your collecting duct decides really what your urine looks like. Is it gonna be clear? Is it gonna be yellow? Is it going to be this beautiful orange that a lot of us see around finals, right? Is it going to be bubbly? Like, what is it going to look like? And your collecting duct is really the one that tailors it at the very end, okay? So you tailor a lot of different things. One of the main jobs of your collecting duct that you want to remember is that it helps with pH regulation, okay? And what's interesting with pH regulation is that alkalosis, being too basic, this is where we regulate it, is the nephron, right? So alkalosis is regulated here. We can't regulate it with our breathing because when we breathe out acid. There's no way to breathe out base. So the only way that we can remove base from our body is through the kidneys, okay? I would highly suggest that you make flashcards of this table, because this table is really helpful with the big patterns that you see in the nephron. Okay. All right. Any questions about what we're going to see in the next few slides, or are you guys good? If it's amber, you're really dehydrated, you should drink some water. Yeah, good job. All right, so we looked at reabsorption. It'll come back, I promise. The other thing that we're going to look at is secretion. Okay, so secretion is removing different items from your blood and putting it into the filtrate. And when you think about secretion, you're really doing active transport. You're pushing it out of your blood. You have to get it from that side to the other side. Okay. So some things will require ATP, some things will require secondary active transport. But the key here is that you need to move it from one side to the other. You definitely have to move it from one side to the other because it didn't come out as filtrate. So it's a large item. So let's see, large or charged. So I'm going to put that here, large or charged. All right. So most of secretion occurs in the PCT. So you notice that the PCT does a lot of stuff, right? So it absorbs a lot of material, and it's also going to secrete some of the big stuff into our filtrate. Okay. One of the main ones is like drugs. So everyone always asks that. Drugs are going to be secreted in the PCT. Okay. The other thing that matters to us is going to be some of our favorite ions, right? So potassium is one of them. Remember that we really tailor the amount of potassium because it affects our neurons and it affects our muscle. So our PCT is so vital for that. So imagine if your kidneys don't work, right? If you have renal failure, you're going to have trouble adjusting potassium. Okay. So you're going to have trouble absorbing it, right? And secreting it. So it's important to monitor it. Another one that's really important, and I'm going to put a heart on it, is H+. H+ is acid. Okay. So we have acid-base regulation in the PCT, but we're not regulating base, we're regulating acid here. Okay, so that's important. If we need to remove acid, we're going to do it here. Okay, if we're going to keep acid, we're going to slow down the secretion of H+. Okay, the last one is NH4, right? So that's what gives you that pee smell, that ammonia smell. Okay, other ones are creatinine. And creatinine is, I put it in there because that's one way that we're able to determine how your kidneys are functioning. Okay, so we can see if they're functioning well or not as well. Okay, last but not least, I think we did talk about that. Last but not least, they can make bicarbonate ion. Okay, so these cells are not going to be removing it from the blood directly, but they can make it and then secrete it out of that cell. Okay, so acid-base regulation. Okay. All right, so let's look at secretion. Some of the main reasons that you care is number one, drugs and metabolites. Okay. Other things are plasma proteins. So if you have too much of a certain type of plasma protein, you can go ahead and move it from the blood into the filtrate through tubular secretion. Okay. That's going to take transcellular transport. Okay. The other things that we can do is we can remove things like urea and uric acid, right? I don't care too much about that, but I do care about K+. Okay, so K+, remember, we highly, highly regulate it. So if we have too much K+ in our system, then we're going to go ahead and secrete it out in the PCT when we have a certain hormone, okay? And that hormone is going to be aldosterone. So aldosterone is going to help us regulate the amount of K+ that we have in our body, okay? But I want you to remember that this is happening in the PCT, okay? Cool. All right. Last but not least, we always talk about this, but acid-base regulation in tubular secretion is important, right? We can move these, but they're ions, so we need to secrete them out if we need to remove them from the body, okay? When you look at this picture here, it's a good one to study. This is a good summary of what gets secreted where. So you can see the blue is how we reabsorb items, and the green is how we secrete items, okay? So you can see how we're secreting H+ and some drugs. We're secreting K+, right? In a few places, we can secrete urea, right? But the key here is that when you're secreting, remember that this is going to be active. We're moving things that are charged or large, and we're trying to get rid of them from the blood. When you're reabsorbing, right, that again, if it's large and charged, then you're going to have to use energy to move it from one place to the other. If it's small, then you can go ahead and reabsorb it through paracellular transport, okay? So those are your big things that you want to remember. So what we want to look at right now is how we reabsorb our favorite things, which is water and salts. Okay, so that's your main goal, water and salt. We looked at glucose already. Glucose, super important. Water and salt are the next ones. So we need to know about gradients. Okay, so what I'm going to do is I'm going to go over what's on the left. Okay, and then I'm going to give you a break because we're going to come back and you're going to have a lot of different small things that you have to remember. Okay, so I'm going to give you a break so you have a fresh mind. So on the left here, you have a little bit of what we went over in anatomy. So you have your medulla. Remember that the medulla is salty. You have the cortex that is isoosmotic. So here the cortex is 300, right? Isoosmotic. The medulla will go from 300 to 1200. Okay, so I want you to remember that. The cortex is 300. The medulla will go from 300 to 1200. Okay, it's salty, salty. The other thing that you want to remember is that the loop of Henle goes into that medulla. So it's going to go in and it's at 300, 400, 500, all the way down to 1200. Then it goes back up 1100, 800, 500, right? Up until it gets to the 300 area. Okay. So I'm going to go ahead and give you guys a 10-minute break. So return at, let's see, 3:43, like at 3:55. Okay. So break. Okay, when you guys come back, then we're going to start going into this. Okay. But I want you to have a fresh mind because it's a lot of small little concepts that sometimes feel overwhelming. Okay. So we'll try to break it down. All right. I'll see you at 3:55. If you have questions, go ahead and ask. If not, then I'll see you soon. And I see Karen asked some things. So let me see if anyone else. About 60. You also have phosphate being taken into blood, right? Phosphate, yeah. Yep. Yep. You have a lot of different things. But we're only going to focus on the main ones that matter to us, right? So mostly water, the 4 different ions, right? So potassium, sodium, chloride, and which one am I missing? Sodium, chloride, potassium, calcium. Those 4 we really care about. Okay. And then sugar was the other one. You have a lot of other things too, right? Like what we see here. You can see there's a bunch of other things like urea, NH4. You have how different drugs are transported, but we're not going to go too in depth, right? we're focusing on those 6 things first. And then we're going to focus on how they are reabsorbed or secreted at different sections of the nephron. Okay. Cool. But yeah, there's a lot of stuff. It's really complicated, but it's actually really, really fun. It's I think it's a lot of concepts that you already know. You're just putting them into this system. So the hardest part, I think, for this is actually when you're looking at something like, let me see, like this, right? So one of the things that's sometimes skipped is understanding where these proteins are, right? So is this the apical or basal side, right? It's the apical side. So the apical side is next to the lumen. So it's important that you remember that the sodium glucose transporter is on the apical side and the sodium potassium pump is on the basal side, right? Next to the interstitial fluid. The glucose transporter is next to the interstitial fluid. So those are small things that you want to try to remember, not so much for this class, but like when you take pharmacology, you're going to want to know like what side is each one of these on, because you want to know how the medicine is acting on the different transporters. Okay. Let me see. Yeah. So like where they start and where they are going and why. Yep. You're right on track. Yep. And like all of these things that we're learning right now, you're going to see them again when we go to fluid electrolyte balance. So it'll come back. Everything that we're just setting up the problem. So we're setting up like, this is the cell and here are the proteins. And then the next time we come, this is the cell with the proteins. This is how the hormone affects it. Okay, so it'll just keep coming back. That's a good question. Thanks for asking. All right, so have fun, take your break. All right, so hopefully you guys are mostly back. All right. Righty. All right, so what we want to do is we want to go through, I guess I should go back this way, is go through how the loop of Henle works. Okay, so your main goal is to figure out how water is reabsorbed and how salts is reabsorbed. Okay. All right. So we're going to be looking at the descending and then the ascending limb. Okay. So some key things to remember is that the descending limb is where we reabsorb water. And the ascending limb is where we reabsorb salts. Okay. So always remember that descending water, ascending salt. Okay, descending water, ascending salt. And remember that solutes suck. And they suck water, right? So when we're moving salt into the nephron, or sorry, into the medulla, that's gonna add the solute to the interstitial fluid. And the more salt we pump out, the more water we're gonna reabsorb. More salt, more water. So it's a cycle, right? The more salt we pump out, the more water we reabsorb. Okay, so that's your main pattern. That's what you're seeing in this little loop here. It shows you that the more salt we pump out of the filtrate, the higher the osmolarity in the medulla and the more water we can reabsorb, the more water we suck, right? Solutes suck. And that's basically how this works. So we're going to look at the next figure on the bottom and we want to see how the gradients affect it. Okay. So remember that things will always, they will move, or water will move to the area that has the most amount of solute until it reaches equilibrium, right? So that's what we're seeing here on the figure. So blood is 300 milliosmolarity. Okay, so the osmolarity of your blood is 300. That means that the plasma is 300. So when we take the plasma and we move it through those filters, right? So through the fenestration, through the basement membrane, and through the podocytes, when it comes out to the other end and it gets into the Bowman's capsule, the osmolarity is also 300. So 300, 300. When we start off in the proximal convoluted tubule, we're still in the cortex, okay? So we're gonna keep our 300 status. So you notice that we stay at, whoops, we stay at 300, okay? So 300 in the plasma. When it becomes filtrate, we're at 300. When we get into the proximal convoluted tubule, we're gonna maintain that 300. Okay, and all of this is in the cortex. Okay, the cortex is 300. So what happens as you go down into the medulla? As you go down, the osmolarity changes. So it gets higher and higher. Okay, so as you go down, you have more solute, and the more solute you have, the more water you suck. Okay, and that's why you see water being sucked into the medulla. Okay, so we're going to draw this out on this slide just to give us a little bit of space. Okay, so I'm just going to show you how it works. You don't have to draw this if you don't want to. That's okay. Because I'm going to erase a lot. Okay, so remember that we start off at 300 in the proximal convoluted tubule, right? We're at 300. We're going to go down into the nephron, or down into the loop of Henle, and then we're going to be at 400, right? So this is going to go down. It's at 300, and the outside is at 400. Remember that solutes suck. So you have 400 outside, 300 inside. You're going to move water from the 300 to the 400. Solutes suck. So water is going to move in this direction, okay, until you reach equilibrium. So there's going to be a point right here where we reach 400. Okay, so now we move to the next part. Let's say that now we're at 600, right? We're at 400 and then 600. Solutes suck. So you're going to go ahead and move water towards the outside. Okay, so we're going to move water this way until you reach equilibrium. So now we're at 600. We're going to go down one more time, right? And we're at 1200. Solutes suck. So you're going to pull water out until you reach equilibrium, right? And equilibrium here is 1200. So we're going to go to 1200. Does that make sense how the pattern works? Okay, so it's all about increasing the osmolarity on the outside so that you continuously suck water into the interstitial fluid. Okay, and that's important because you are reabsorbing a lot of the water. You're preventing dehydration. Okay, that's why that part is really important. This part here helps prevent dehydration. Because you don't want to lose 1% of your water every time you pee. That would be really bad. So you have to reabsorb as much as you can back into your blood. Okay, so remember that anything that goes in here goes back into your blood. So now let's go ahead and turn the corner. You have 1200 here at the bottom. Now we're going to go up and we're going to go into the ascending limb. Now, when you look at the ascending limb, the main thing you want to remember is that you're actively pumping salt. Whenever you hear the word active, you're going to be using energy. Okay. So here we're actively pumping salt. Okay. And that takes energy. And when you think about pumping, you can make a gradient. So anytime you hear the word pump, you can make a gradient. Okay, and what kind of gradient do you think we're making here? Are we making a water gradient or a salt gradient? What kind of gradient do you think we're creating? Salt. Okay, so that salt gradient is what causes water to be reabsorbed. So if that salt gradient isn't being made, you're not going to reabsorb water. Okay, the higher the gradient, the faster the rate. So if you have a low gradient, low rates. Okay. The other thing that's important here is that gradients are able to move things from high to low concentration and from low to high. Okay, so you can go ahead and make that steep gradient there. So I want you to pay attention to the numbers here, right? We're going to start off at 1200 where there's a lot of salt. We're going to start pumping it out, right? Moving it out of the filtrate. We're going to move some more. We're going to keep moving out more salt. And I want you to see what happens when it gets out of the nephron, or sorry, out of the loop. What number do you see here? What's the number that you see when it comes out of the loop of Henle? What's that number? 100. So when we look at the proximal convoluted tubule, we see 300. When we look at the distal convoluted tubule, we see 100. That's a big difference, right? So we have an area that is hypoosmotic in the filtrate and an area that is 300 outside, okay? So remember the cortex is in 300 land. So we have 300 outside, 100 inside. We made the filtrate less concentrated, right? We diluted it. We removed enough salt that it's less concentrated than what we started off with, okay? So we're really keeping as much salt as we can, okay? So now we're at 100, and that's important because the next part after your distal convoluted tubule is the collecting duct. And remember that the collecting duct can tailor your urine to either be clear, yellow, or orange, right? When you pee out clear, you're close to that 100, okay? You have a lot of dilute urine. When it's more concentrated, you're closer to that 1200, okay? And we'll see that in a minute. But it's important to remember here, PCT, 300. The loop of Henle will go from 300 to 1200, and then 1200 to 100. Okay, so make sure you know those patterns. 300, then you go 300 to 1200, 1200 to 100. Okay, you're reabsorbing water when it goes down. You're reabsorbing salt actively when you go up. Okay. All right, any questions about this down here? Are you guys good? Awesome. All right, so let's look at something called the counter current multiplier. Okay, so this just means that things are moving in different directions or different concentrations when you're going down the tube versus when you're going up the tube, right? So here we're concentrating urine and here we're diluting urine, okay? That's the main thing. You're concentrating and then you're diluting, okay? Cool, that's all this means. So don't get too caught up in what's the difference between countercurrent multiplier and countercurrent exchange, okay? Multiplier just means that you're using the gradient in the medulla to help you reabsorb water as you're going down and reabsorb salt as you go up, okay? When you look at the exchanger, this is a little bit different, okay? So when you look at the multiplier, that's just the nephron. When you're looking at the exchanger, you're exchanging between two systems. So this is renal system and blood, okay? So the main thing to remember here is if you look at how filtrate moves and then how blood moves, we'll have this kind of relationship, right? So filtrate will move in this direction and blood will move in this direction. So the key here is that the gradients tend to be opposite, right? So you're gonna have reabsorption, whoops, the other way. Reabsorption from one to the other by using gradients, okay? So by having opposite gradients along when they're next to each other, you're gonna be able to pick things up better, okay? Don't worry too much about it. Just know that they're running in opposite directions and things will move from high to low concentration and then solutes suck, okay? So if my blood has more salt in it compared to the filtrate, it's going to be able to suck the water back into the blood, okay? And the opposite is true, right? If I have more water and my concentrations are lower, I'm going to suck less water back in, okay? All right, so let's go to the nitty gritty. This stuff you definitely want to know. So the collecting duct is your nitty gritty, right? It is how you tailor make that urine, okay? So remember the key points from our last two slides. Blood is 300. When you make filtrate in the very beginning, in the proximal convoluted tubule, you're 300. When you enter the loop, you start at 300, you go down, down, down to 1200. Then we turn the corner and we go up and we make it more dilute. So 1200 up to 100. So in our distal convoluted tubule, we're at 100. And our collecting duct is going to start off with that 100, and that's important, okay? So I'm going to circle 100 here and here because we're starting off with 100. And in the cortex, we're at 300. Okay, so we have a perfect gradient. If we need to reabsorb items, we can use that gradient. If we need to secrete items, then we can use ATP to move them out. Okay, but here's the key. Your collecting duct could either be very leaky, and that's when it has a lot of aquaporins. And that means you have a lot of reabsorption of water, or it could not be leaky. That means you have a little bit of aquaporin. That means you reabsorb a little bit, so less water, and you pee more. I should put here pee less. Okay, so you have these two different scenarios. On the left side, you have someone that is highly hydrated. Okay, lots and lots of water. Okay, when you have too much water, that's not a good thing because you're diluting the osmolarity in your blood, but you're also increasing the blood volume. Okay, so it's important that we get rid of that water. And one of the ways that we do that is by maintaining this 100 all the way through the collecting duct. Since it's staying at 100, we're not reabsorbing water. Okay, that means that we're in this scenario here. Okay, so we're adding in less aquaporins, we're reabsorbing less water, we're going to pee all of that excess water out. Okay, and that's going to help us maintain our osmolarity, maintain our blood volume. Okay, when you're dehydrated, like someone that's running, okay, and they're in the desert, so you know they're sweating, that person needs to keep as much water as possible because they're constantly losing water through sweat and breathing. So that 100 that we start off with, it's going to change into something more concentrated. And the way that we can concentrate that filtrate is by moving water back into the blood. Okay, so we're going to add aquaporins and we're going to move that water back into blood. So you can see that water is being reabsorbed. And we're going to get down to close to 1200. So we're making it really dilute. Or sorry, really concentrated. So this person, when they pee, they're going to see that orangey color that some of us see, right? When we're dehydrated. The person that's drinking a lot of water here, when they pee, it's going to look really clear. Okay. That's just the difference between trying to remove water versus trying to keep water. Okay. What you want to remember is the relationships that you see here, right? And you also want to remember the hormone that helps you do this. So the main hormone that will affect the aquaporins at your collecting duct is ADH. This is also called vasopressin. Okay, so when vasopressin is present, then you're going to keep water. When you don't have vasopressin, you're going to remove water. Okay, so let's look up at the feedback loops that you see. So let's start off with the person that's drinking a lot of water. When you drink a lot of water, the osmolarity, the concentration of your blood decreases. Okay, so you have less than 300. If that happens, you're going to release out less ADH, less vasopressin. Okay, and that means that you're going to have less aquaporins in your collecting duct. Okay, so now instead of having a lot of water moving through, you have less water moving through. Since you have less water moving through in the collecting duct, you're going to keep a lot of that water in the filtrate. Okay, and when you do that, you're going to go ahead and pee all that filtrate water out when you make your urine. Okay, so it's going to stay at around 100. When you're dehydrated, it's a little bit opposite. So instead of having a low osmolarity, blood osmolarity, you have high blood osmolarity. You're going to release out more ADH, more vasopressin, right? So when you're dehydrated, ADH is present. So you're going to have a lot of it. That's going to tell your cells to add more aquaporins. That means that water is going to be able to cross through super easy. Water is going to be reabsorbed, put back into the blood, and you maintain the water that you have, okay? Remember that the only way you can get more water is by drinking. So here it's about retaining and maintaining any water that you do have, okay? So this person's going to go ahead and pee out that yellow-orange color, okay? All right, any questions on this, or are you guys good? You guys all good. All right, make sure, please, please, please make sure that you know ADH, okay? Make sure you know that feedback loop. Make sure you know it really well for lecture and for lab. The next part here is urination, right? How do you urinate? You probably went over it in anatomy, so we're just gonna go over it real quick. And I'll tell you the key points, okay? The main job of this is to get rid of any urine that you have stored, and then you can go ahead and add more urine to that storage bag, right? The bladder. When your bladder is full, you're gonna have stretch receptors on the wall that will sense that full bladder, okay? It's gonna send information to your body to go ahead and affect the sphincters that are keeping the urine inside of your body. Okay, and remember you have two different ones. You have internal and external sphincters. Okay, internal and externals. The one that you get to control is your external. The one that you don't control is your internal. Okay, so your internal one will go ahead and relax to allow urine to flow through when the bladder is full, whether you like it or not. Okay, so whether you like it or not, it'll relax. You have control over your external, so you can go ahead and keep it closed until you're ready to urinate. Okay. So this is the one that we train. Okay, so when you think about potty training, you're training this one. Okay. Once your stretch receptors are signaled, you're going to increase parasympathetic, decrease sympathetic. That'll cause you to relax. And then the one that you control, external, these are somatic motor neurons, right? This is you telling your body to relax. So instead of having them contract, you're going to ask them to relax, to open up. Once both are open, you can pee. Okay, not bad, pretty easy peasy. And remember that you have control over external, not internal. And stretch receptors are the key. Okay. All right. The next part here is all about body water content. Okay, so how much water do you have? How much solute do you have? And how does that change with time? Okay. So infants are about 73% more water, right, than us. Adult males, about 60%, and adult females, about 50%. Okay. When you look at assigned females, they'll have more fat content, less muscle mass. Assigned male, more muscle mass, less fat. And then little babies, they're going to have lower body fat and lower bone mass. Okay, so water is just everywhere. You change the amount of water as you grow. Okay. When you think about where water is inside your body, I want you to think about this. A lot of your water is in the cell. Okay, this is in cells. Okay, you have a lot of water also around your cells. And then you have water in your plasma. Okay, imagine that you had to go and regulate every single cell. That would be really hard, right? But we can definitely affect what's in our plasma, what's in our blood. And that's why the renal system is important, because you're filtering that blood, filtering that plasma all the time. And the plasma is connected to the interstitial fluid, right? They communicate back and forth. And the interstitial fluid is surrounding your cells. So this communicates back and forth. So if you affect the plasma, you're in turn affecting the interstitial fluid, and in turn affecting the cell. Does that make sense? And if you affect the cell, you affect the interstitial fluid and you affect the plasma. So they're all related one to the other. So part of the reason you're highly regulating the plasma is because you really want to make sure that you're regulating the fluids that are in your cell. Okay, so that's how your body works. When you look at the body fluids, the other thing that matters is solutes. Okay, so we know that waters are big solvent. But solutes are also important. Okay, when we think about solutes and we think about fluids, you have electrolytes. Okay, and the electrolytes that matter are right here. Okay, so you notice our big four, right? They're there. The other one that really matters here is bicarbonate ion, because bicarbonate ion is a buffer system in our body. And the other one is a phosphate group that also helps us buffer. Okay, so when we look at solutes, we also want to make sure we're really regulating it when we're looking at what's in the plasma, what's in the interstitial fluid, and what's inside the cell. So you see blood plasma in red, interstitial fluid in blue, and intracellular in yellow. So if we look here, what you see in plasma really reflects what's in interstitial fluid, right? So if you just look at red and blue, just compare them, right? You notice that they're very closely mirroring each other. So when we test someone's blood, we're really trying to take a look at what their interstitial fluid looks like, okay? So you notice that they kind of mirror each other. And then when we look at intracellular, right, it can differ. So these are the yellow ones. I'm going to put triangles over them. So they can differ a lot between the plasma and the interstitial fluid, okay? And that's because what you put in the cell can be placed there actively, okay? So a lot of times we're only gonna have what we need in our cell, and that's important because the proteins in our cell need to stay working, and if we add too much acid, it would affect it. Too much base, it would affect it. Too much protein, it would affect it, okay? All right. I think we talked about these already, so I'm going to leave this alone because we already talked about it. One thing I do want to mention, though, is this here, okay? When we look at someone's electrolytes and we're trying to see whether they're within normal range, we're going to be using milliequivalents per liter, okay? And this is the equation that we use for that. I'm not going to make you use the equation, right? But I did want to bring this up so that when you take chemistry and you get to the point where you have to calculate milliequivalencies, you need to pay attention because this will help you read your blood panels. It'll help you calculate how you're going to make your IV bags. And then all these other things when it comes to drugs, right? Dosage. So just make sure that you pay attention to that, okay? All right. So here we want to just remember that what you have in your plasma affects what's in your interstitial fluid, and that affects what's in your cell. Okay. So we want to move the items properly from one side to the other. So we have a few examples here. The first one is in the lungs. We want to get our oxygen. We put it into blood. That then goes into the interstitial fluid of the target, and that goes into the cell. Okay. When it gets used up, you make CO2. CO2 gets placed in the interstitial fluid, back into the blood, and we remove it out of the lungs. Other things are things like nutrients. We bring them in through the GI, right? We absorb them into our blood. Those nutrients go everywhere and our cells use it. The same with ions in water, right? You bring it in, then they use it, okay? But you can see with all these different systems, we're constantly just moving things back and forth, okay? So we're constantly monitoring the gases, the ions, the water, okay? All right, so one of the key things that we want to understand, right, is how we balance water. So think about it, water, ions, glucose, right? All those things we want to learn how to monitor it, okay, and balance it. So we want to know how we balance water. So the way that we get water into our body is by drinking, okay? That's the only way. You have to drink, must drink. Okay, we make some through metabolism, but not enough to survive. Okay, so we make a little bit through metabolism, but not enough to survive. You have to drink water. Okay, so small amounts. We can lose water in lots of ways. Some of the ways that we lose water is through sweat, right? Other ways is through speaking. Other ways is through secretions, right? Like mucus secretions. And other ways are through urination, okay? Every time you poop, you lose a little bit of water. So if you're dehydrated, your poop is drier than normal. If you're really hydrated, your poops are smooth, okay? So if you're struggling with pooping, you should drink some water. Another thing that we wanna remember is that when we're talking about water and water consumption, it could be a glass of water, but it could also be water in anything that you drink or eat. So sometimes when someone's dehydrated, they can give you things like Jello, because Jello has a lot of water in it, okay? Soup has a lot of water in it. I always bring in like all these cultural references, right? Think about when it's hot. What does your mom usually give you when it's really hot? And you're like, why are you giving me this? They usually give you soup, right? When it's really hot weather, traditionally, a lot of cultures will give you soup. And the reason why is because they're trying to have you replace water in your system. And the other thing they're trying to replace is salt. So it's not out of nowhere. It's actually a really smart idea to give you a lot of soup. All right. So let's look at what happens when water is not balanced. Okay. The key to water is osmolarity, right? That's one of the main things. So osmolarity. You want to keep it at around 300, right? Remember 300 is your favorite number, 300, 300, 300. So if it's too high, that's going to be a problem. If it's too low, that's going to be a problem. Okay, so if you have a rise in osmolarity, that means that you've lost water or you have too much salt, okay? So the first thing is getting water back into your system. And remember that the only way you can get water into your system is drinking. So your brain is going to tell you that you feel thirsty and you're going to go look for water. Okay. The other thing that it's going to do is that it's going to cause you to release out vasopressin. You want to keep water. So you're going to release out ADH. Bring it back, bring it back, bring it back. Okay. If you have a decrease in osmolarity, then you usually have too much water in your system, okay? In this case, you're not gonna feel thirsty, you're gonna feel fine, right? You don't need more water. And you're gonna go ahead and stop secreting out ADH. Oops, oh man, it doesn't let me underline it. So you're gonna inhibit ADH. So instead of keeping water in your body, you're gonna pee it out. You're going to release it. Okay. So let's take a look at the feedback mechanism for water. Okay. So here we have osmolarity as our stimulus. Another stimulus that can trigger this though is plasma volume. Okay. So we have two scenarios, high osmolarity and low plasma volume. Okay, so we're going to start off with high osmolarity. So if you have a high osmolarity in your blood, okay, that means that you are too salty, right? Too much solute in your blood. That's going to cause osmoreceptors in your hypothalamus to sense it. So you have osmoreceptors, they sense the osmolarity in your blood all the time. And they're going to cause you to feel thirsty, okay? The way that that works is that you have these cells in your hypothalamus that will shrink when the osmolarity is too high. And if it shrinks, it tells your body that you're thirsty, okay? The other thing that happens is you have low saliva and dry mouth. And your hypothalamus can sense that you have low saliva and dry mouth, and it's going to make you feel thirsty. Okay. When you get that signal from the hypothalamus, it's going to cause you to want to drink. You're going to go look for it. You're going to drink, and hopefully when you drink, you moisten your you moisten your mouth. And you're going to go ahead and moisten the rest of your GI that's also dry. Water is going to be reabsorbed all throughout the GI. And as the water is being reabsorbed, it goes into your blood, okay? When it goes into your blood, then that's gonna dilute the osmolarity of the blood and it'll decrease the osmolarity, okay? The other thing that can trigger it is plasma volume. So if your plasma volume is too low, okay, you have low blood volume, that's gonna cause your blood pressure to go down. Okay, if your blood pressure goes down, then you're going to have granular cells in the kidneys, right? Remember granular cells from the Juxtaglomerular apparatus? They're talking to the macula densa. So if you have low plasma volume, you're going to have less filtrate and your macula densa is going to sense that low filtrate. It's going to tell the granular cells, hey, we have low filtrate. We got to do something about it. So your granular cells are going to secrete out renin. Okay, that renin is going to go into your blood, and it's going to cause the RAS system to activate. Okay, so that one of the key things with the RAS system is that angiotensin 2 is going to go up. Okay, that's another hormone. And angiotensin 2 is going to help you regulate the amount of salts and the amount of water that you have in your body. Okay, so you notice that it comes right back to your hypothalamus. Okay, so angiotensin 2 is going to tell your hypothalamus to make you feel thirsty. You're going to feel thirsty. You drink some water. You drink water, you absorb it, and it's going to decrease your osmolarity and increase your plasma volume. Okay, so one thing to remember is that you're seeing two different feedback loops here, right? One for osmolarity and one for volume. And they come out with the same result at the end, right? They're both going to do this, but two different stimuli. So my question to you is, will this work if you only have a change in osmolarity? Will this feedback work if you only have a change in osmolarity? So let me change that question. Will you have this response if you have a change in osmolarity? Will you have this response if you have a change in osmolarity? Good job. Will you have this response if you have a change in plasma volume? Will you have this response if you have a change in plasma volume? Yeah, also yes. Okay, so you can have this happen. That's a possibility. Okay, and in your lab, you saw this. When you eat chips, you increase the osmolarity in your blood, but you're not adding more water. You're just adding salt. So you see this happen. This can also happen all by itself. Okay, so this is what happens if, let's say, you get cut, right, and you're losing blood, right? This is going to happen. Okay, so it's not like you're adding salt. It's not like you're removing salt. You're just losing plasma. Okay, so this is like when you're shot, when you're cut. Let's say you had a baby and you're hemorrhaging. This is what would happen. Okay, this can also happen, right? Where you have both of these things happening. Okay, and this is what happens when you're dehydrated. So when you're dehydrated, you're sweating off a lot of water. So your blood plasma goes down, right? And as you're losing water, the osmolarity goes up. Okay, are you guys good with those three examples? Your lab really, really helps. And none of the annotations stayed. That sucks. I'm going to try to recover it, but I got it. It's a good day. All right. So one of the things that you want to remember, right, is that you want to know all the pieces of this feedback loop, right? So stimulus, sensor, controller, etc. Okay, so make sure that you can go through and really understand the stimulus, sensor, and controller. Okay, one of the things that you're going to notice when you do your lab or when you did your renal lab is that we try to be very clear about when your blood is changing in osmolarity or volume and when your urine is changing, right? Blood is your stimulus. What's happening in your blood is going to stimulate what happens in the renal system. Urine is your response. Got it? Blood, stimulus, urine, response. So what's happening initially is changes in blood, right? That's what you see here. And what happens at the end of the day is resulting because you made urine. So if we look at this feedback mechanism and we think about what's happening, we're going to change the way that we make our urine in order to have these results. Okay. So we're going to have that in our next slide. So this tells us how we change the urine. Here, we're just talking about thirst. Here, we're looking at how we're tailoring our urine to keep as much water as we can. Okay. So we're seeing some of the same stimuli: increased osmolarity and decreased plasma volume. So the stimuli is the same. Okay. So increased saltiness or decreased volume. So let's go through what happens when you have increased saltiness. Okay, increased osmolarity. Your osmoreceptors in the hypothalamus will sense that because they shrink. They're going to tell the posterior pituitary to release out ADH. Okay, so hypothalamus is going to get that information. It's going to tell the posterior pituitary to, or it's going to send, ADH to the posterior pituitary, and it's going to release it there. Okay, remember that ADH is made in the hypothalamus, and these long axons go from the hypothalamus to the posterior pituitary. So you're going to go ahead and release it out. It's going to go into the blood, and blood goes everywhere, right? When it gets to the kidneys, ADH is going to bind to receptors on the collecting duct. And the key thing that happens there is that you're going to add aquaporins, because so ADH goes to the collecting duct. The collecting duct adds aquaporins. And if you add aquaporins, you can move water back into blood super easy. Okay, and that's what we see. We're reabsorbing water. So we're taking water from the filtrate and putting it back into the blood. That means that our filtrate is going to have a lowered amount of water. Okay, and that's what we notice. We notice that we have lowered urine volume because we're moving the water back into our blood instead of peeing it out. The other result is that the movement of water back into our blood is going to help us decrease that osmolarity and increase the plasma volume. Okay, so make sure that you remember how ADH works. Make sure that you remember where it's made, where it's secreted. Make sure that you remember that osmo receptors are in the hypothalamus. And then make sure you remember those results: increased aquaporin, increased reabsorption. You make less urine, but you get to keep more water. So plasma volume goes up, osmolarity goes down. Okay. If we look at the other feedback loop that's here, this one's based on plasma volume. So you have lowered plasma volume. When you lower it, then you're affecting blood pressure, right? So blood pressure also goes down. If blood volume goes down, blood pressure goes down. And in this case, the feedback mechanism that is going to be triggered is the baroreceptor reflex. Okay. So should you review the baroreceptor reflex that we drew a few weeks ago? Yeah. You need to review all of those steps of the baroreceptor reflex. Okay. The baroreceptor reflex is going to sense the low amount of blood pressure, and it's going to trigger the release of ADH. Okay, so you're going to release out ADH from the posterior pituitary. ADH is going to go to the collecting duct. You add more aquaporins, you reabsorb water, you keep more of your water. That helps you increase your plasma volume, and you end up decreasing urine. Okay. So when you look at this and you think about this pathway, this is your long-term effect. and baroreceptor is your short-term effect. Okay, so if you have changes in blood pressure, your baroreceptor can change the circumstances really quick, right? But anything that has to do with the renal system, it's going to take a little bit longer. Okay, so it's not going to be seconds, it's going to be like half an hour, right? It's going to take a while before you see the changes. Okay, all right, so some reasons that you might see the changes are listed here. We talked about a few, but make sure you go and review it. Okay. All right. Any questions about ADH or your thirst response? I'm going to leave you guys here. So you guys okay? All right. So when you guys come back, we'll look at dehydration a little bit more. Okay, so I would recommend that you just go through these slides and just kind of refresh your memory. Okay, most of this you already learned in your lab. So a lot of this is just kind of repetition. Okay, all right. So have fun. I'll see you guys when you come back. And we'll continue with fluid and electrolyte balance. Okay. Have fun. If you have questions, stay on. If not, have fun. Oh, I didn't see these questions. Hold on, let me see. Why do you have a filter going out of your room? I don't know what that means. Like, oh, that's my air conditioner, just in case you're looking. Yeah, I live in a really old house with no insulation. So when it gets hot, I'm dying. And when it gets cold, I'm also dying. So we have a lot of heaters and ACs that really help. Daniela's asking, our grade is based on the grade scale in the syllabus, not the grade, the Canvas grade, right? It should be going right along with what you're doing in class. So one of the key things to remember is that the grade scale is tentative, which means that it may be subject to change. So because we're removing the reproductive system, it's going to change a little bit. So at some point when I get to that and I work on those assignments, you'll see those changes. But it's pretty much the same, right? So if you look at the pie chart that is on your syllabus, that's about how your grades are split. So remember in the very beginning, we said, be consistent in your work. So do very well in your homework. Make sure that you're consistently doing well in your lab quizzes. And if you can do that, that's half your grade. And then the other half is your exams. So your key job for your exams is learning how to pass your exams. If you can learn how to pass them and get an average of a passing grade, then you're doing very, very well. Okay.